Hello, my name is Janet Lorv, and I'm a PhD candidate from the University of Waterloo. And today I want to present to you my work on gene defragmentation in long-read assemblies using reference-based error correction. So to kind of start things off, as some of you may know, long-read sequencing is becoming an increasingly popular form of sequencing. And one of the main reasons for this is that assemblies generated from these reads are highly contiguous compared to their short-read counterparts. However, there is one big main caveat in that these assemblies are more error-prone. And why that is important is that many of these errors, which are primarily insertions and deletions, can introduce frame shifts and premature start and stop codons in a gene. In fact, a lot of annotated genes in these assemblies are likely to be fragmented. So what can we do to address this issue is actually we developed a software tool that is aimed to detect and correct these types of errors. And the idea behind the software is to actually use publicly available reference information to inform the tool on where errors might be in the draft assembly. And this can be done using both reference genome information as well as reference protein information, depending on what type of data that the researcher might have on hand. And an overview of this workflow can actually be seen in Figure 1. So what essentially happens is that the software takes in 3 inputs, 2 of them mandatory, 1 of them optional, and starts an error detection phase, followed by the optional error adjustment phase. And once the detected errors are finalized, the software then uses the detected errors to correct the draft assembly. So to kind of go into some detail into the software, how we actually detect errors actually differs based on what information is given. So for, say, reference genomes, the software uses an alignment difference between the reference and the draft assemblies and calculates what is known as an ADS, which is an alignment difference frequency for every single error type and every single position. So for example, like in Figure 2, every single of the implied insertion is used to calculate the ADF for a candidate deletion error. Although there is one thing to note is that indels greater than 2 nucleotides will be ignored, and that's because we consider them more to be strain differences than actually assembly errors. So once the ADF is fully calculated, the software passes through the assembly in a set window size and evaluates each region for a combined ADF of 1, meaning that there is some consensus across all the reference genomes that there is a candidate error. So if you want to look at Figure 2, this actually region wouldn't be considered to have an error because one of the reference genomes actually disagrees with the other 10. So for proteins, on the other hand, the software uses aligned protein fragments to detect the likely candidate error sites. The idea here is that an error is likely to be contained within an interfragment region when the alignment, the fragments are actually not in agreement. So to kind of implement this idea, what we did was we took the top scoring pair for each of the proteins as a seed to approximate the location of a potential gene. However, the lower scoring fragments that also lie within the same expected region and are on the same strands are then grouped together and merged. The software then evaluates the interfragment regions between each of the fragments for a candidate error. Once we find all the candidate errors, we then use raw read information to determine if the candidate has resupport. If there is resupport, we can then determine whether we want to retain or adjust the candidate error based on the same alignment difference principle that we used in the detection stage. So after error adjustments, the errors are then finalized and used to correct the draft assembly. So to test our software, we actually collected 11 equally distant reference genomes that hovered between 94 to 98% identity to our assemblies, and approximately 5,000 ref-seq proteins from the Pseudomonas syringae species, and both of these data was aligned to our draft assembly, one of which was generated solely from nanopore sequencing data, and the other assembly is just a typical Illumina draft genome. The genomes were then aligned in a semi-global manner with LASZ, while proteins were aligned to the assemblies using T+N. So after running the software on our 2 assemblies, we were actually able to detect less than 3,000 errors, meaning that we are correcting less than 0.05% of each assembly. And if you look at Table 1, with both detection methods, we were able to find substantially more errors in the long-read assemblies compared to the short-read equivalent. However, there is still some potential errors that were found in the Illumina assembly. Unsurprisingly, the majority of the errors for the protein detections were within the genes themselves, and the genome-based errors were actually kind of dependent on the assembly, with the long-read assembly containing more gene errors and the short-read assembly containing more intergenic errors. So to confirm the correction of these assemblies can actually improve the assembly quality, we annotated each of the assemblies both prior and after correction using PGAP. As you can see in figure 4, overall the number of complete genes increased in the long-read assembly for both detection methods and did not really change for the Illumina assembly. In fact, the Illumina assembly is very slightly improved with a few genes being annotated post-correction. If you look under the two types of fragmented genes, actually, the majority of the improvements in the long-read assemblies were from the reduction of frame-shifted genes, with some of the incomplete genes showing improvements as well. So with the software, we were able to actually reduce the number of fragmented genes from 23.3% to 5.6% in the long-read assemblies, which is nearly comparable to the Illumina assembly's 4.4%. We also looked into the errors we were correcting and found that the long-read assembly, the majority of the errors were detected were associated with long homopolymers, as shown in figure 5. In fact, most of these homopolymers were adenines and thymines, which is consistent with the type of errors seen in previous studies. So in conclusion, we were able to develop a software tool that can use homology information to detect and correct common errors found in long-read assemblies. And then the correction of less than 0.05% of the assembly can drastically reduce the number of fragmented genes to a level comparable to short-read assemblies. And lastly, this software may also improve short-read assemblies. So thank you for listening, and I would like to briefly acknowledge that this work was made possible with NSERC, and thank you again.